Hello everyone, my name is Andrea. I am the Regional Marketing Specialist from GenScript. Today's topic for the webinar session is Tips for Successful ELISA Kit Development. And I'm very excited to introduce you the presenter today, Dr. Pei Liu. Dr. Liu is the Product Manager of Reagent Antibody Services at GenScript. She graduated from Nanjing University with a PhD in pharmacy and joined the GenScript in 2019 and served as Senior Scientist in Antibody Department. Her work is mainly engaged in antibody development and the establishment of single B-cell platform. In addition, she worked with various cases and have accumulated a wealth of experience on the development of the ELISA kit. We are currently running an ELISA kit beta testing program, and please check out our website and enjoy further price markdown for your custom ELISA kit project. The information will be also included in the email followed with our webinar, along with a recording of today's webinar. Today's webinar will take around 45 minutes. If you have any question, feel free to type in the question field you see on your screen, and we'll answering them after the presentation. If we didn't get to answer your question during the Q&A session, we'll be sure to take down an email back to you afterward. We will record down this email webinar, which will be available on our website after today. Lastly, here's a list of upcoming webinars that we will be rolling out in the following weeks. Without further ado, let's welcome Dr. Pei Liu to start her presentation. Dr. Liu, over to you.
Okay. Thanks for your introduction, Andrea. Hi, everyone. This is Pei. I'm happy to be here to share with you my experience of the ELISA kit development. Here is our outline of our topic. We will go through principles and general types of ELISA, then the application of the kit. Most important, most important, I will introduce how to generate an ELISA kit and how to troubleshoot ELISA assay. We will also review how GenScript generates ELISA assay for our customers. Finally, some cases will also be shared here. As we know, ELISA were first developed in the early 1970s as a replacement for radioimmunoassay. The ELISA is distinguished from other antibody-based assay because the separation of specific and non-specific interactions occurs via serial binding to the solid surface, usually a polystyrene multi-well plate, and because quantitative results can be achieved. The ELISA procedure results in a colored end product which correlates to the amount of analyte present in the original sample. So it's quick and simple to carry out, and since they are designed to rapidly handle a large number of samples in parallel, they are very popular choice for the evaluation of various research and diagnostic targets. Here, the basic ELISA procedure involves plate coating, blocking, washing, and signal generation and measurement. In general, the ELISA has four formats. First is the simplest format, direct ELISA. First, the direct ELISA, the antigen will be immobilized in the well of an ELISA plate. Then the antigen is then detected by an antibody directly conjugated with the HRP, with the enzyme HRP. Direct ELISA detection is much faster than other ELISA techniques because fewer steps are needed. The assay is also less prone to error since fewer reagents and steps are needed. No potentially cross-reacting because there is no secondary antibody needed. Although there are some disadvantages to this method, as the antigen immobilization is not specific, Higher background noise may be observed in the comparison to indirect ELISA. This is primarily because all proteins in the sample, including the target protein, will bind to the plate. Directly, ELISA is less flexible since a specific conjugated primary antibody is needed for each target protein. Also, there is no secondary antibody is used, so there is no signal amplification, which reduces assay sensitivity. So the direct ELISA technique is typically used when the immune response to an antigen needs to be analyzed. The second format is the indirect ELISA. The same with the direct ELISA. First, the antigen will be coated on the plate. A primary antibody binds to the specific antigen. Then an enzyme conjugated secondary antibody directed against the host species of the primary antibody is applied. The indirect ELISA method has a higher sensitivity since there are more than one labeled secondary antibody can be bind to the primary antibody. It's more economical than the direct ELISA because fewer labeled antibodies are needed. Indirect ELISA delivers greater flexibility since different primary antibodies can be used with a single labeled secondary antibody. Among its disadvantages is the possibility of the cross-reactivity of the secondary antibody to the absorbed antigen, which could increase the background. Also, the indirect ELISA assay takes longer run than direct ELISA because it has 3 steps. The indirect ELISA is the most suitable for determining total antibody concentration in samples. The second format is the sandwich ELISA. It's very widely used in our ELISA kit development. The procedure for sandwich ELISA firstly requires a well of an ELISA plate to be coated with the capture antibody. Then the antigen in the sample will be added in the well. Then during the stage 3 step, the paired another primary antibody will be added in the well, and finally the secondary antibody conjugated with the enemy HRP will be added. The key advantage of the sandwich ELISA is higher sensitivity. It's about 2 to 5 times more sensitive than direct and indirect ELISA. It also delivers higher specificity as 2 antibodies are used to detect the antigen. It offers flexibility since direct and indirect methods can be used. The advantages brings with them a few advantages. First, if a standardized ELISA kit or test antibody pair is not available, antibody optimization has to work out since it's important to reduce reactivity between capture and detection antibodies. Sandwich ELISA are particularly suited to the analysis of the complex samples since the antigen does not needed to be purified in the assay, so it can deliver the higher sensitivity and the specificity, and the sample can be human serum and mouse serum, horse serum, and so on. Finally is the The final format is the competitive ELISA. Is also known as a blocking ELISA. Perhaps this format is the most sorry. This format is the most complex of all the ELISA techniques. Each of the above assay types can be adapted to a competitive format. In this format, first the antigen will be coded on the well, and another unknown antigen will be added in the well. If there is a higher concentration of antigen in the sample, the significant reduction in signal output will be observed. In contrast, if there is very little antigen in the sample, there will be very little reduction in the expected signal output. So its main advantage is there is no sample processing is required, and the crude or impure samples can be used. So to summarize the three, there's 4 types of the ELISA. Both they have their advantage and disadvantages. I have talked with you about the slides, so I will skip these slides. As we know, ELISA is used for the detection and quantification of antigens, antibodies, and other molecules. Besides application in the clinical diagnostic, ELISA is used as the exclusive assay method for many research purposes, such as characterizing new proteins and developing new drug therapies. For therapeutic drug, we can develop PK and ADA and HCP ELISA kit. Also for the food safety, it's very important. It includes a number of routines that should be followed to avoid potential health hazards. Food safety consideration include the origins of food, including the practice relating to the food labeling, food hygiene, food additives, and pesticide residues, and so on. For the agriculture diagnostic, we will testing the soil, water cysts, and plant tissue samples, and so on. Ensure the safety of the samples against the hazardous contaminants such as pathogens, toxins, and heavy metals, and so on. So for the animal and plant disease diagnosis, they will use diagnosis of the animal plant disease and research on biotic pathogens and host pathogens, and so on. So although many in vitro diagnostic companies manufacture reagent kits for research use, they can't keep pace with the ever-expanding needs of researchers investigating new proteins. So how could we generate an ELISA kit in-house? What's the key parameters needed to be optimized? Here are the answers. First, you should clearly about the detection limit required, the sensitivity. Sensitivity is the capability of the method measuring the analyte at a sufficiently low concentration for the intended application of the test. Second is the specific. The kit should be present in the samples. In the specific, there should be no cross-reactivity to molecules structurally similar to the analyte that may be present in the samples. Another is simple. The kit should be easy to perform and gives quick results. The kit should be also stable and safe. The reagents used in the assay are thermostable and the analytical performance is robust. The reagent components used in the method are not harmful and require no specific handling. Except the 4 parameters. For different ELISA kits, we need to pay attention to different parameters. As we know, the detection of an analysis of ADA formation is a helpful tool in understanding potential immune response. Information on immune response observed during the clinical trials particularly in the incidence of ADA induction or any implications of ADA response affecting the PK and the safety and the efficacy is crucial for any therapeutic protein product development program. So detection of the ADA formation is highly dependent on the sensitivity and the specificity and the drug tolerance level of the assay. Also, the assay count point is the level of the response of the assay that defines the sample response as positive or negative. So, establishing the appropriate count point is critical to minimize the risk of false negative results. We should consider the myriad of the interfering products or matrix components. So FDA recommends examining assay drug tolerance early in the assay development. Also the sponsor may examine the drug tolerance by deliberately adding different known amounts of positive control antibody into the ADA negative control samples in the absence or presence of different quantities of the therapeutic product to determine whether the therapeutic protein product interferes with ADA detection. So the drug tolerance may be improved using approaches such as the acid dissociation that disrupt circulating ADA drug complexes. So the selectivity is very important. It's the nature of the target and the type of the positive control should be taken into consideration when developing the assay because these factors impact the estimate of the drug tolerance. For the HCP ELISA kit generation, the antibodies ability for the coverage parameters is also very important. For PK, we should consider the selectivity and the dilution effect. Now we will go through the troubleshoot for ELISA assay. Sometimes we have weak or no signal observed the false negative results. Maybe the reagent was not at the room temperature before assay. So we should allow the reagents sit in the room temperature for 15 to 20 minutes until completely thawed. So if we don't incubate the plate for a proper time or temperature, we should increase the incubation time or follow the manual to repeat the assay. Sometimes maybe we use the wrong buffers. And we also can sometimes we have a poor standard curve. So you should we should pay more attention on if the wells are not washed or aspirated properly, or if there is bubbles in the wells, or the incomplete reagent mixing, and so inconsistent reagent preparation. As we know, the majority is about 19% of problems observed in the practice of ELISA are operator flaws caused by lack of common sense, fear to appreciate the need to stick to the instructions, or sloppy technique or poorly maintained equipment. So how GenScript generates the ELISA kit? Here is our workflow. As we know, GenScript have more than 15 years experience for antibody generation. We proudly offer hundreds of ELISA kits in both sandwich ELISA and competitive ELISA. At GenScript, we have generated many kits for our customers. Here is an example how we generate the ELISA kit. When the project come in, we will design and prepare the immunogen, and after immunization and fusion, we will get many ELISA positive clones. We will do the affinity ranking and epitope binning, and we will select the top 5 clones to do a small scale production. Then we will conjugate these antibodies with the enzyme HRP or biotin for pairing. Finally, we will select the top 3 best paired antibodies for the kit's development. If the clients need more ELISA kits, we will make a manufacturing for our customers. For antibody generation, we have 2 platforms. Here is our customer Hybridoma platform. Is the most traditional and mature method. On this platform, we can generate mouse and rabbit monoclonal antibodies. Here is our milestone and our timeline. Another important platform is the single B platform based on Beacon. From the comparison, you can see that Hybridoma platform mimics some B-cell repertoire, but not for Beacon. And also, the single B platform can shorten the screening time from 12 weeks to 1 day. This is the biggest advantages. 先停一下，我嗓子好像有点难受。So based on the platform, we have generated COVID-19 N protein and S protein antibodies. We have both Hybridoma platform and we got the cell line from the Beacon. We got the antibody sequence. We also have the rabbit antibodies with higher affinity. They can be used for WB ELISA kit development and ACE2 blocking assay. Here, these are S protein antibodies from mouse based on the Hybridoma platform. We have got the IgG antibody and the IgM antibody. We have tested the affinity and the epitope. Both IgG and IgM have a higher affinity and diversity. These are S protein antibodies from the rabbit. Based on the based on beacon platform. These antibodies have a higher affinity and also diversity. Often, and the affinity constant determines the potential sensitivity of the assay. It's recommended that the affinity constant be greater than 10 to the 10 or 11 power, and ideally is 10 to the 12 power. Most rabbit antibodies are more than 10 to the 11 power, so the affinity is very high. Then these are M-protein antibodies based on the Hybridoma platform. Some clones also have the higher affinity. Then we conjugated the antibody with biotin and acquired 8 paired antibodies. We choose one of the best antibodies for our ELISA kit development. As we know from COVID-19 diagnostic, they include the COVID-19 disease diagnostic and SARS-CoV-2 virus detection. For COVID-19 disease diagnostic, there is included the routine blood test for the increased liver enzymes and muscle enzymes and myoglobin. They are infection markers in blood. For chest imaging, we will do the multiple small patchy shadows and the extrapulmonary bands and so on. Another important part is the SARS-CoV-2 virus detection. SARS-CoV-2 virus detection includes the molecular testing. They include the real-time PCR and the NGS. Those that detect the sequence, the target in the sample for RNA. Another is for the antibody and protein detection. As we know, detection of antibodies can be used both for diagnosis and population surveillance. Antibodies test how many people have had the disease, including those whose symptoms were minor or who were asymptomatic. An accurate mortality rate of the disease and the level of herd immunity in the population can be determined from the results of this test. For virus protein detection is also a method to monitor the infection state. So we have developed 3 kinds of ELISA kit for SARS-CoV-2 virus detection. Here is the details of the project workflow for general ELISA kit. There are 4 parts. For milestone 1, we will prove of the concept. We will identify best pairs and evaluate the performance in sensitivity and specificity. Select the best pair antibodies for sandwich ELISA and evaluate the consistent data with customers' organization. For milestone 2, for the assay development, we will prepare and validate the assay standards and test the sensitivity and specificity. Milestone 3, we will manufacture 5 kits for the assay validation. Validate the stability, accuracy, and variation of the assay kits. Also, the criterion of the assay kits for the intra-assay and inter-assay. Finally, we will manufacture the immunoassay kits in 96 test formats in GenScript. Here is the SARS-CoV-2 N-protein ELISA kit. We use the sandwich ELISA format. From the 8 paired antibodies we choose. This is the workflow. So from the 8 paired antibodies, we choose the 3H11 and C11 and the 4H2G1 antibody used. One reason is that they has a good linearization. Here another reason is we use the health human mix serum to test the matrix effect. It's not affected by the matrix, so we also optimize the optimize the reaction time. The whole test can save 1 hour. Normally, this incubation time is 1 hour. Here is 1 hour, so we saved about. Here is another is about 30 to 1 hour. We in our format we can save the whole test can save 1 hour time. The fundamental objective for any ELISA development is first we will identify the correct antibody or antigen. Second is create an optimal environment for assay kinetics and binding affinities. For the third is we provide consistent results with maximum assay sensitivity and specificity. So to meet these objectives, we need to balance many factors. Sensitivity and precision are key goals. So after optimization of the coating concentration, reaction time, detection concentration, the sensitivity of the kit could be 30 pg/mm. Precision, also called the accuracy, is another important parameters. As we know, precision is a measure of the variability in a series of measurements for the same material run in the method. Results should be reproducible within and between assay runs to assure adequate precision. To provide reliable estimates, we evaluate both the intra-assay and the inter-assay. Our tests are better than the FDA requirements. Sometimes the FDA required is about 10%. We are higher than their requirements. Stability is the validation studies of determining the analyte stability after freeze and thaw cycles, and after short-term and long-term storage. All stability experiments should be performed on samples that have been stored for a time that is longer than the actual storage period. So stability is evaluated by analysis of at least 3 replies of core QC concentration level before and after stability storage. The mean accuracy in the measurements at each level should be within 20% deviation of the theoretical concentration in principle. Acceleration in a 37-degree incubator is equivalent to a 40-degree storage is about for 1.5 months. So this is what we often call the Arian equation. Our kit can be stable for 1 year. Here is a summary of the COVID-19 and protein ELISA kit. Our kits have a good performance with a higher sensitivity and linear range and a higher precision. Here is our kit log number. Here's the kits tool. We developed the SARS-CoV-2 RBD IgG ELISA detection kit. We use it's also called the capture ELISA. First, we will coating the anti-human IgG in the well and blocking the well, and we add the test sample mixed to the wells. If the sample have the antibody, it will bind on the primary antibody. When we use the detection antibody here the detection is the RBD protein conjugated with HRP. Will bind to the RBD IgG. Then we will use the substrate to catalyze the enzyme to generate the colored readout. IgG antibodies to SARS-CoV-2 generally become detectable at the 10 to 14 days after infection. They may be detected earlier and normally peak around 28 days after the onset of infection. So we have tested 62 clinical samples. They are different patients from different infection states. So the detector results is variable, but our kit performance is very well. Both the sensitivity and the specificity are good. So here is a summary of the SARS-CoV-2 RBD IgG ELISA detection kit. We use this maybe you can call the capture ELISA. It had a good performance with a higher sensitivity. This is for clinical samples detection results. The sensitivity could be 90% and the specificity could be 100. Also, we have a higher. We also have a higher precision. Another according to the immune response to infection, as we know at the infection of earlier time, IgM antibodies are generally detectable in the blood several days after the initial infection. Although levels over the course of infection are not well characterized, so IgM detection is very useful for earlier detection. The principle is the same as the IgG kit, so I just show the detection results. Here is our positive control. Here is the clinical detection results. As you can see, the patient detection results is very different. Maybe here the patient 16 is at the earlier infection status. Here the summary of the SARS-CoV-2 RBD IgM ELISA detection kit. We also called the capture. The method is the capture ELISA. For the 62 clinical samples, also we have a higher sensitivity, about 88%, and for specificity is 100%, and the higher precision. We also have a higher for the intra-assay and the inter-assay. For GenScript, we offer service that of all the different ELISA kit formats. In in addition, we also offer the specialized sandwich ELISA kit development services such as the PK and ADA and HCP ELISA. And we know for the PK, it can quantify the serum and protein drug levels to provide accurate PK data to guide the optimization of the drug dosing regimens. And for the host cell, we'll evaluate the presence of undesired host cell proteins during the purification process of the biologics. So this is our an overview of our advantages for antibody generation. For kit development, the antibody is very important. So here is our advantages. We have optimum antigen design tool. Using this tool, we can generate superior peptide antigen sequence. And we also have the Immune Plus. It can break the immune tolerance of highly homologous antigens. Also, we can provide the gene immunization service. GenScript offers a wider array of custom antibody service. However, besides our services alone, we offer some amazing advantages to help make your research easy. In fact, we have generated so many higher quality antibodies that we even award the most popular customer antibody supplier of 2018 and 2020. On top of our experience, our services offer a wide array of patented technologies to increase efficiency of your antibody. So most importantly, we often offer milestone-based project management consultation and pricing with every customer project. These are our customers. They are famous biological companies and pharmaceutical companies. So okay, everyone. I have at the end of our presentation, and now we would like to move the QA session. Okay. I have to answer the question. Andrea?
Yeah, thank you, Dr. Lupe, for your wonderful webinar. So let's go through several questions from the attendees. The first question is, difference between the COVID-19 disease detection and SARS-CoV detection, is there any difference procedures to follow?
Yes, it's a good question. For the disease diagnosis, is mainly based on the clinical diagnosis. So virus detection is a tool to verify you have infected the virus. So the booster tests need to be performed simultaneously.
Thank you very much. So the second question is, can you please explain how do you name antibodies?
It is an interesting question. As we know, we used the 196 well place. Text we selected the paired antibodies 3H11C11. For example, 3 represents the order of the place we led. As you know, we will plate 14 place for our customers. The H presents the rules. The 196 well place have H rules. And the numbers 11 represent the number of the wells. As you know, they have in the row they have 12 wells. For the C is we will do the subclonal for our selected antibody. So is standard the first subclonal time we selected from the place C. In the row is C, and the 11 number represents the number of the wells. Did you understand how to name our antibodies?
It could be a little bit difficult for us to understand, and if there's any question, please leave down. So let's move on to the third question. Were there any possible biotin interface?
After labeling, dialysis will be done, so there will be no effect of the biotin interference.
Thank you very much. So the next question is, can SARS-CoV-2 end application in magnetic microbead immunoassay?
We didn't try to these antibodies used by the microbeads, but I think it should be possible. If customers need, we can provide antibodies for our customers to test.
Thank you very much. So the next question is, hi, for the SARS-CoV-2 RBD IgG ELISA kit, how we ensure that the entire well is able to capture all different types of IgG in the sample? Because in the human sample, they are lossy words. Can you please answer that question?
This is a very good question. We have taken this issue into account when developing the kit. So we optimized the concentration of coated antibodies to ensure that more antibodies can be captured and verified with the clinical samples. As you can see, we do a lot of clinical samples. The sensitivity and the specificity are very good. So there is no interference of different types of IgG effect in the sample. If you use our kit, it's not a problem.
Thank you very much. Move on to the sixth question. So for case study 3, may I ask what's the cutoff to differentiate between a healthy and infected patient? Because based on the data, some detection value of the patients, and then I missed the word there. Can you please answer the question regarding to case study 3?
For the differentiate from between healthy and infected patients, our cutoff is 1.16. Is the sensitivity as I showed in the slides for the IgG kit is about 90%. Clinical samples will be detected is consistent with the clinical results. For the IgM, it's about 88%. So I think our kit performance are very good. It can be used in the clinical samples detection.
Thank you very much. Move on to the next question. How about the sensitivity of ELISA kit for COVID-19 detection?
I have answered the question. The sensitivity is about. Did you say the N protein ELISA kit?
Sorry, maybe I missed that part. So maybe let's move on to the next question. Could you classify sandwich ELISA as a point of care diagnostic?
Yes, the sandwich ELISA, as I said, the sandwich format is more sensitive, precise, and robust. So it can distinguish antigen from impure samples. The difficulty is it can it may be difficult to generate an antibody paired which recognize the different epitopes. So to be honest, sandwich format is the most used in our during my career. We have provided many services for our customer. The most widely used format.
Thank you very much. Move on to the next question. Do you produce antibodies derived from B cells and or plasma cells of human origin?
I'm sorry, could you repeat the question?
Sure. So do you produce antibodies derived from B cells and or plasma cells of human origin?
Human origin. We produce the antibodies from the plasma cells, not from the mouse and the rabbit, not from the human.
Thank you very much. Yeah, sorry. Keep going.
But we can do ceramic antibodies. We can make the humanize our antibodies by use the recombinant method.
Thank you very much. Next question is, which automated instruments have your COVID-19 kits been validated on?
Which, I'm sorry.
Sorry.
The signal is not well.
So the question is, which automated instruments have your COVID-19 kits been validated on?
Do you type the question? I'm sorry, I can't listen clearly.
Sorry, can you hear me?
Yeah.
Okay, let me repeat the question once again. Which automated instruments have your COVID-19 kits been validated on? If you can't really hear me, hello, Dr. Liu.
Yes, maybe I can answer this question after
later on. Yeah. So thank you everyone for joining us. I know we have still quite some questions haven't been have the time to answer. So we will come back to you guys with a separate email after this webinar. The webinar will be recorded and we will send out a follow-up email to let you guys know about where you can review the webinar recording and come back to you also with the answers for the questions. So thank you very much for joining us today and have a good one. Thank you.
Oh, thank you.
Thank you.
Bye.
Bye.